Okay, so remember we're talking about, oh, video 2, sorry guys. Remember we're talking about chromosomes, and we're going to talk about how we need to redistribute them when we go through this cell division thing. So remember your vocab, big on your vocab. You've got a lot of words that are going to be really similar. Okay, so as we're getting ready for cell division, the chromosomes are going to start to condense. And what that means is during prophase, which we haven't talked about yet, but during prophase, all that DNA is in loose strands. It looks like a bowl of spaghetti. There's a bunch of spaghetti noodles. And intermixed within those noodles are these small little histone proteins and some other proteins, but the ones we talk about most are histones. And those histones are like meatballs. So this is like spaghetti and meatballs. Okay, so what's going to happen is we're going to start to see that DNA start to wrap and wind around the histones, and they're going to start forming those nice little condensed X's that we're used to when we talk about or when we draw chromosomes. Okay, so when it's loose and spaghetti bowlish, the DNA is called chromatin, and then it starts to condense and we call it a chromosome. Because it replicates, because it duplicates, that chromosome goes from being a single condensed strand, and it copies itself, and we have two copies. So we have the original and we have the copy. So we have two sister chromatids. Okay? But they're connected to each other. They're connected by something called a centromere. Well, they're connected at the centromere. The centromere is the narrow spot or the narrow waist, and there's some protein there that's going to bind the two halves together. It looks something like this. Okay, so we started out with a single chromosome, all condensed. It underwent DNA duplication. It replicated. It copied itself, if you will. And now I still have a single chromosome, but it has twice the DNA. It has twice the genetic material. I know that's confusing. We need to talk about that when we meet. So somebody remember to ask me that. And then here is the centromere. So the centromere is the center part. It's where they connect. And this centromere is just here in this drawing. It can be up, it can be down. The centromere can move. And we'll see that in the karyotyping lab. And then we're going to undergo mitosis. And what's actually going to happen is we're going to split. The centromere is going to pull apart, and we're going to end up with two separate chromosomes again. So that's what we're doing here. So mitosis is the division of the nucleus. Cytokinesis is the division of the cytoplasm and all of the organelles. Gametes, the egg and the sperm, sorry about that. Gametes, the egg and the sperm, are produced through a different process. They're produced through something called meiosis or meiosis. That's our next chapter. Okay, so we're not even going to really tackle that here other than to say at this point that in mitosis, I'm going to get genetically identical daughter cells. In meiosis, I'm going to get daughter cells, but they are not genetically identical, and they actually have half of the chromosomes, half of the DNA is the original. All right, there you've got a whiteboard question. I'll count to 7, and then I'll flip it over and give you the answer. If you've got wiki sticks or pipe cleaners or popsicles, spaghetti noodles, anything like that that you can use at home, those will be helpful. Okay, so there was a German anatomist named Walter Fleming, and he was the first to actually see mitosis, to be able to document mitosis and cytokinesis and what was going on. A couple others get credited as well, or at least we're seeing it independently of Fleming, but he's the one who actually typically gets the kudos. All right, so we got to talk about cell cycle. Remember, cell cycle was the life cycle of the cell. So we're going to be made up of interphase, which is just a growth and copying sort of stage. We're going to further break down interphase. Vast majority of the cell's time is spent in interphase. And then the one who gets all the limelight, though, is the mitotic phase. And the mitotic phase is mitosis and cytokinesis. Okay, so that's the actual nuclear and cytoplasmic dividing. Interphase, take a peek at this. So interphase is made up, it's about 90%. Okay, so it's all of this. It's interphase, all of this with this big outer greenish arrow. This is all interphase. And interphase is about 90% of the cell cycle. And we divide it into 3 subphases. We divide it into G1, S, and G2. Okay. So I just want to peek a second. I want to see if I did those differently for you. No, so I'm going to do them right here. Okay. So G1 and G2, you can think of, they're called gap. They're called gap phases, which is not a great way to try and remember them. I think of them as growth. Okay, I think of that G as for growth. So I have this initial growth phase where the cell is growing, the cell is undergoing all the changes it needs to potentially start DNA synthesis. And then it moves into the S phase, which is the synthesis phase. That should be easy to put together. And this is where DNA replicates. Okay, so this is where we're going to duplicate the DNA and go from one chromosome into that chromosome with 2 sister chromatids. And then we finish out the growing and the preparing for mitosis, replicating what organelles we can, increasing cytoplasm. Sorry about that, another sneeze. So increasing the cytoplasm, doing all of that. Now the thing I dislike about this imagery is that I love that it divides it. Don't get me wrong. It's great that it helps us get G1S, G2. Those are all fabulous things. It helps us to see that interphase is the majority of the cell cycle, roughly 90%. But the thing I don't like about it is that it leaves us with the impression that growth, 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 and then all growth stops, and we do this DNA thing. And then when the DNA thing is done, now we go back to just growing. And it doesn't actually work like that. Yes, growth slows down during the S phase, but it's still running, it's still going. So it's kind of like those programs that run in the background on your computer. They're there, they're doing what they need to do, they're just not doing it as efficiently as if nothing else were going on. Okay? So, and then our last phase is mitotic phase, which is mitosis and cytokinesis. We started with 1 cell, we go all the way through this process, we end up with 2 cells. So that's what that imagery is showing you. Lots of different books will divide mitosis differently. And the big point of contention is they don't use prometaphase. Back in the dark ages, when your favorite biology professor was learning this, we didn't have prometaphase. We just had prophase, and we called it early prophase and late prophase. So if I do that, if I revert, I'm sorry. Late prophase is the equivalent of prometaphase. I think prometaphase is easier for you guys to remember because it's halfway between prophase and metaphase, so prometa. But it's also what your textbook, it's also what Campbell uses in all of their most recent editions, so that's what we're going to use, is we'll use prophase, prometaphase, metaphase, anaphase, telophase, and then cytokinesis. Cytokinesis actually begins somewhere in mid to late anaphase when we look at all the chemical markers and stuff, but again, your text isn't great at it. Texts disagree significantly about where to stick cytokinesis. I just say if you can recognize that it's well underway by telophase and it's wrapping up close to the end of late telophase, I'm okay with it. I'm good with it. It's close enough. I don't think we know the answer, so it's hard to hold you to a concrete answer when we don't actually know the concrete answer. This is an overview, and I've got it a little bit bigger version for you to see here, but this is what we're going to do. So we're going to go, this is G2 of interphase, then we're going to go prophase, prometaphase, metaphase, this is probably the easiest for my students to recognize, anaphase, and then telophase with cytokinesis. So here we go. What you're going to want to do, what you're going to want to do is you're going to want to know these things, you're going to want to know the order, and you're going to want to know key features all the way through. What's happening here, what's happening here, what's happening there. Whether you learn it as a bullet point for me or you learn it as a continuous paragraph story, I don't care. you know, I'm comfortable either way. I can grade them either way. You need to understand this process and what's happening along the way. Okay, so G2. So in G2, so that last of the growth phase, right? So we're right here. Okay, so second gap. Okay, this is G2, this is second growth, this is where we are. Okay, we've already replicated, we've already duplicated our DNA. We have 2 copies of every chromosome. Okay, here we are. So here I have this 2 copies of every chromosome. This thing here is the nucleus. Here's the little nucleolus where we make ribosomal RNA. Here are 2 centrosomes. These guys are going to move, okay? We'll talk about them in a second. Our nuclear envelope is intact. Our plasma membrane is out here. All this brown stuff is the cytoplasm and the organelles. They just didn't draw in the organelles. Okay. This right here, where we have this giant bowl of spaghetti, that's called chromatin. Remember, I have 2 copies of every chromosome in there at this point. In prophase, a bunch of stuff is going to happen. So in prophase, these centrosomes, okay, these guys are going to start to migrate. They move from being here, just kind of hanging out close to the nucleus, and you can't see me moving my hands, but I'm moving them to opposite sides, and they're moving to opposite poles of the nucleus, or of the cell, sorry. We're starting to get some early microtubule formation coming off of there, some spindle fibers that are forming. We're also starting to build some little itty-bitty protein fibers, little itty-bitty microtubules off of the centrosomes. And what they're going to become is they're going to become the asters. And the asters are going to be the anchors that hold this centrosome in place when it gets to its end location. That's all outside the nucleus. Inside the nucleus, what's happening is we're starting to see this chromatin. It's starting to wrap around the histone proteins, and we're forming these nice little neat condensed chromosomes that we're used to talking about. The sister chromatids will stay together. So what we're going to have is we're going to have these sister chromatids. So I have 2 copies of chromosome 1 from dad and 2 copies of chromosome 2 or chromosome 1 from mom, and so on and so on and so on. So I've got all of this DNA going on. In prometaphase, so as I'm making the transition into metaphase, we're going to see we've got these condensed down, so they've finished condensing. Okay, and the other thing that's happened is our centrosomes have moved to opposite poles, opposite sides of the cell. The asters are coming off and they're anchoring those centrosomes, because what's going to happen is we're going to start pulling things apart. We've got these microtubules that are coming out from the centrosomes and the centrioles, and they're moving towards the opposite side. We need to talk about those again in a second. The nuclear envelope is breaking down. Oh, something I forgot to tell you. The nucleolus is completely gone by prophase. Okay, it actually just disappears from view. It disintegrates essentially. Okay, so nuclear envelope is fragmenting, is breaking apart. It's actually being recycled at this point. Now, I said we had to talk about these microtubules, right? So there are 2 types of these microtubules. There are the kinetochore microtubules and the non-kinetochore microtubules. This is another good visual for us in class, so somebody remind me about that. But kinetochore microtubules actually come up here and they attach at the centromeres. At the centromeres there are these little proteins. Remember I said there were proteins holding these together? Those proteins are called kinetochore proteins. So the microtubules will come one from this side and attach to the protein on this sister chromatid, and one will come from this side and attach to the kinetochore of this sister chromatid, and now I've got a method for separating these 2 sister chromatids. The other type of microtubule are the non-kinetochore. So if kinetochore attach to kinetochores, non-kinetochores don't attach to kinetochores. They actually come and they'll overlap each other. So we're going to get an overlap of these microtubules, these spindle fibers. You can use those interchangeably, by the way. We're going to get an overlap of these spindle fibers across here. Okay, so what's going to happen is these guys are going to actually pull apart and shorten, and these guys are actually going to push off of each other. And then that's prometaphase. So we're setting up. Notice we're also seeing that these guys are moving towards a sort of a midline plane of that cell. So that at metaphase, nuclear envelope is entirely gone, centrosomes are completely anchored at one pole, kinetochore microtubules have all attached, non-kinetochore microtubules are overlapped, and then everybody is lined up on this plane. Now there's an image right here. There's this image, and I'm going to pull it right here because I need to remember to tell you that, see how these guys aren't in a straight line, they're in a plane. Remember that that cell is three-dimensional. Every time we draw it to look like this, and they're all lined up, like ducks in a row, like soldiers in a parade, like, I don't know, for those of you who organize your Skittles in a line by color before you eat them. However, whatever you can think of that's organized in a nice, neat, orderly fashion. Pickets on a fence, if you will. Okay? And it's not like that. It's a plane. It's a three-dimensional thing. So they're not all lined up end to end to end to end to end like bricks in a wall. They're all in this one plane. Okay? So they're staggered a little bit. There's not enough space for them to all line up end to end to end. Okay, so that's metaphase. And that's the key thing about metaphase, is that they've just all come together. They're lined up at the metaphasic plate. Sorry. The spindle fibers are all attached. Kinetochores are connected. Non-kinetochores are not. And then in anaphase, we're going to start to see those migration of those tubules. And what's going to happen is, here at the centrosome, where these centrioles are, we're going to start ratcheting in, and we're going to start pulling back these microtubules. We're going to start separating these sister chromosomes. And once we get them fully separated, we don't call them sister chromatids anymore. We actually call them daughter chromosomes. Okay, so we go from being sister chromatids when we're attached to being daughter chromosomes when we're not attached. Okay? I know, confusing. Sister chromatids, daughter chromosomes. Remember, this whole thing right here, these two sister chromatids together, that was one chromosome. Now if I come over here to the third one, I don't have sister chromatids anymore. I have daughter chromosomes, and this is not one chromosome anymore, but it's actually two separating chromosomes. If that's confusing, ask me in class. We'll go through it again. But anaphase is just the pulling apart, and then we're going to come to telophase and cytokinesis. In telophase or telophase, either is fine. What we're going to see is we're going to see disintegration of the microtubules in the spindle apparatus. We're going to see reformation of the nuclear envelope. We're going to see the nucleolus reappear, reform. We're going to start to see the chromosome. Remember, these are daughter chromosomes. We're going to see the daughter chromosomes start to unwind. They're going to go back to a chromatin state. We're going to see a division of the cytoplasm and the nuclei. And here's your hallmark for knowing, if you're looking at a microscopic slide, here's your hallmark. When you start to see this indent in the side of the cytoplasm, the cleavage furrow, you know you're in telophase. So if you're kind of in this ifty mode and I don't know where I am and I can't really see and I can't really tell, look for the cleavage furrow in animal cells. In plant cells, you're going to look for the formation of the cell plate, and we'll look at that in a minute. But that's how you're going to tell telophase. Okay, I'm going to pause this one. We'll publish this video, and then we're going to go look at specifically at the spindle fiber.